Good evening. Welcome to Pandemic Perspectives, Week 3. If this is your first time joining us, my name is Amy Finley, and I'm the Dean of Students at the University of Michigan-Dearborn. I am so pleased to welcome you to tonight's presentation, and welcome back to everybody who has been with us the first 2 weeks. Tonight is the third part of our exciting 6-part series, led by faculty from a variety of academic disciplines. So far, we've explored the impact of race on care and the example of the pandemic in London in the 1800s. If you missed it, please feel free to go to our Pandemic Perspectives website to check out previous videos. Reminder that at the conclusion of tonight's presentation, we will open up to a question and answer period. Please note that you can submit questions throughout the presentation using the Q&A button at the bottom of your screen. I'd like to introduce you to tonight's presenter, Dr. Raji Janakiraman. Dr. Raji Janakiraman is Lecturer 2 in the Department of Natural Sciences at the University of Michigan-Dearborn. She has been at UM-Dearborn for a decade and has taught courses in molecular biology, microbiology, and epidemiology. She received her PhD in molecular, cellular, and developmental biology from Indiana University Bloomington in 1998. Her postdoctoral training extended from 1999 to 2001 at the University of Michigan, Ann Arbor. She has always been an innovative instructor, adopting active learning techniques, and recently was awarded a UM-Dearborn Creative Teaching Fund. She was also selected to be an AP Biology Reader in 2020. She has a wealth of experience in community outreach, coaching several Science Olympiad events at the elementary and middle school levels, and as a head coach for the Elementary Science Olympiad. In her spare time, Raji likes to cook and go for long walks with her friends. And now, let's move on to our presentation. Dr. Raji, I turn it over to you.
Good evening. I'd like to thank Paul Draus and Amy Finley for giving me this wonderful opportunity. Welcome to Week 3 of Pandemic Perspectives. Epidemiology of COVID-19. What do we know about the distribution of cases of COVID globally, in the US, and even closer at home in Michigan? How has this pandemic evolved? And what do we know about the pathogen that causes COVID-19? I hope to provide you with answers to these questions today. Here is an overview of our agenda for today. I will begin by telling you a little bit about the virus. It's a coronavirus that causes COVID-19. Over the last 2 decades, there have been 3 major outbreaks caused by coronaviruses. Why are coronaviruses so dangerous for us? We will look at where this COVID-19 outbreak originated. I will briefly touch upon the symptoms and complications of COVID-19 and the modes by which it can be transmitted. We will look at how this outbreak evolved to become an epidemic and then a major pandemic. What kind of tests are available to diagnose COVID-19? And finally, at the end of my talk, I will give you an update on the status of vaccine development. SARS-CoV-2, the coronavirus that everyone is talking about. As you all are probably aware, COVID-19 stands for coronavirus disease that was identified in 2019. It began in a city in the Hubei province of China called Wuhan. Doctors were alarmed because towards December of 2019, they started noticing a rapid increase in the number of pneumonia cases, and they did not know what was causing this pneumonia. Eventually, scientists identified that it was a virus, and this virus has been named as SARS-CoV-2. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome, Coronavirus 2. On the left here is a model of the SARS-CoV-2 virus. We're all fairly familiar with this model nowadays. And you can see the characteristic spikes, the crown-like spikes on its surface. That is why it is a coronavirus. SARS-CoV-2 is the third coronavirus to cause a major outbreak. The other two outbreaks that were caused by coronaviruses were SARS, or severe acute respiratory syndrome, and MERS, Middle East respiratory syndrome. We will talk about these outbreaks later. So we keep hearing about viruses. Flu is caused by a virus. The common cold is caused by a virus. And now they tell us COVID-19 is caused by a virus. What is a virus? Viruses are microscopic parasites. They are much smaller than bacteria. Viruses are usually in the size range of nanometers, anywhere from 20 to 300 nanometers. You cannot see them with a regular microscope. You need a special microscope called an electron microscope to view viruses. Are viruses even living? It's debatable. Outside of a living cell, a virus is inert. It has no capacity to reproduce itself. In order to reproduce, it needs a cell. It cannot produce anything without a living cell. How about the shape of viruses? There are some viruses up there on your slide, models of viruses. You can see that viruses can look like tubes or spheres or even alien spiders. Viruses pack light. All they contain is some sort of genetic material. It could be either DNA or RNA, and that genetic material is wrapped up in a protective protein shell, what we call as a capsid. Some viruses, that's all the structure that is, right? These are polioviruses and rhinoviruses that cause common colds. Yet other viruses, like influenza and the coronaviruses, are enveloped viruses. What does that mean? That means outside this protein shell, they have another layer, a fatty envelope. The fatty envelope is derived from the cells that they infect, the host cells. Why is hand hygiene so important to control influenza or even COVID-19? That's because when we wash our hands with soap, we are actually disrupting the envelope of the virus and killing the virus. Coronaviruses are a large family of common viruses that are found in humans, animals, as well as birds. They are so named because when you look at them under the electron microscope, you see that they have a crown around them, or a crown-like spike. So on the left-hand corner of your slide, you can see a transmission electron micrograph taken with a special electron microscope. And you can see spherical particles of the virus, and you can clearly see those spikes that are poking out. What is that spike? It is a protein that is sticking out, protruding from the envelope of the virus. Coronaviruses are enveloped viruses that have single-stranded RNA as their genetic material. On the top, you have a model of the coronavirus, fairly familiar to all of us. You can see the spike protein extending out in yellow. And then the other, on the right-hand side, you can see inside that model, and that pale pink coil you see is the single-stranded RNA, or the genome of this virus. On the top here, you see another type of a picture taken with a different electron microscope called a scanning electron microscope. This scanning electron microscope picture has then been colorized. So you see the SARS-CoV-2 viruses shown to you in yellow, and they're emerging from cells that have been cultured in the lab, and those cells have been colorized blue and pink. From such pictures, scientists have figured out that the diameter of the SARS-CoV-2 virus is somewhere between 60 to 140 nanometers, right? And when they zoom in, they can see crown-like spikes on its surface. The SARS-CoV-2 virus is most closely related to the SARS virus, the SARS-CoV virus, which is the virus that caused SARS, or severe acute respiratory syndrome. It is also closely related to the MERS-CoV virus, the virus that caused another outbreak called as Middle East respiratory syndrome. So how does SARS-2 get into your cells? So again, the spike protein is going to play a very crucial role, we're going to see. And in fact, it is this spike protein that is the target of most of the vaccines that are being manufactured today. The spike protein binds to a receptor that is found on the surface of your cells. What is this receptor? It's called an ACE2 receptor, and that stands for angiotensin converting enzyme 2 receptor. Once it binds, kind of like a key fits into a lock, then there is another enzyme called a protease that chops off half the spike protein. And so you can have fusion between the virus and your host cell membrane. So the insides of the virus get into your cell. So this is called direct entry. Another way by which the virus can get into your cells is called endocytosis. What this means is that the entire virus is swallowed up by a patch of your membrane and pulled into the cell. Once the virus is inside your cell, it takes over the molecular machinery of your cell and creates more copies of itself. The ACE2 receptor is found in several tissues of our body. It's not surprising that you see ACE2 receptors in your respiratory system, in your nose, upper and lower respiratory tract. We know that coronaviruses attack our respiratory tract, but ACE2 receptors are also widespread in heart tissue, in kidneys, in the bladder, even in the eyes. So this particular SARS-CoV-2 virus has the potential to infect a wide variety of tissues in our body. So we have known about coronaviruses for over 50 years. They're not new to us. In fact, 7 strains of coronaviruses infect humans. 20 to 30% of all common colds are caused by coronaviruses. So we did not think of coronaviruses as being highly dangerous to us, highly pathogenic to us, until 2002, when there was an outbreak of SARS, or severe acute respiratory syndrome, in a province of China called Guangdong. This SARS epidemic extended from about November of 2002 till July, so for about 9 months. It eventually became a pandemic. It spread to 29 countries, and 8,500 people came down with SARS. The mortality rate for SARS was about 10%. 10 years after SARS, there was another pathogenic coronavirus that emerged. This was the MERS-CoV virus that caused the Middle East respiratory syndrome. Middle East respiratory syndrome afflicted about 2,500 people. 80% of the cases were localized to Saudi Arabia, but it did spread to other countries. It had a very high fatality rate. Fatality rate was estimated to be about 34%. So why is it so many of these coronaviruses are causing large outbreaks, epidemics, and even pandemics? Yeah. There are multiple species of coronaviruses that are circulating in the wild among different animals that may interact with one another. So this gives these coronaviruses more time to evolve and to recombine. In fact, it is known that coronaviruses have a high frequency of recombination. What does that mean? That means that different coronaviruses can exchange bits of genetic material. And so, voila, you can create a novel coronavirus. It's not surprising given that it recombines a lot. Coronaviruses are respiratory viruses, so they're transmissible fairly easily. The most common mode of transmission is by what is called as droplet transmission. That is, all of us emit respiratory droplets when we cough, sneeze, talk, sing, or even breathe. When you come in contact with these respiratory viruses, you can get exposed to coronavirus. Coronavirus also can spread by direct or indirect contact. Indirect contact is where we have what are called as fomite transmission in epidemiology. What that means is that inanimate objects like a desk, a cup, a pen, on which the virus has landed can harbor this virus. And when people touch those objects and then either touch their mouth, their nose, or eyes, they can get exposed to the virus. That is indirect transmission. So you have high frequency of recombination, easy transmissibility, and the third factor that is so important for these viruses to cause large outbreaks is that the population is highly susceptible. We haven't seen these novel coronaviruses before. These are new to our immune system. And so we are not immune to these viruses at all, and we can get disease as a result. So where are these coronaviruses emerging from? COVID-19, SARS, MERS are all zoonotic diseases. What that means is that they are infectious diseases that are caused by pathogens, in this case viruses, that have jumped from an animal to a human. So scientists have compared the genomes of coronaviruses found in many animals. And let's talk about SARS and MERS first, because we know a lot more about those outbreaks. We are still in the process of this outbreak. So in SARS and MERS, they figured out that those coronaviruses that were causing SARS and MERS were actually coming from bats. The reservoir host was bats. So they were being maintained in bats. And then they spilled over to susceptible humans, probably via an intermediate host. For SARS, the intermediate host was found to be civets, which were sold in the wet markets in certain parts of China. And for MERS, the intermediate host was found to be dromedary camels. What do we know about COVID-19? Where did it emerge from? Scientists have also figured out that COVID-19, or the SARS-CoV-2 virus, is probably coming from the same reservoir host. It's coming from bats, and it has made the jump to humans. Does it have an intermediate host? There was some suggestion that it might be pangolins, but this is unclear at this point. When was COVID-19 first detected? So the outbreak itself happened in mid to late December in Wuhan, China, which is a city in Hubei province. Once scientists knew which virus was causing COVID-19, they did some investigations, epidemiological investigations, and traced back. And they found that the first instances of coronavirus may have been as early as mid-November in China. And so patient 0, who is the first person to have contracted a disease, patient 0 might have very well have been a 55-year-old man from Hubei province who contracted COVID-19 in mid-November, almost a month before the outbreak started in Wuhan. All right, I'd like to show you a brief video that tells you a little more about SARS-CoV-2, the virus, and a little more about the disease, COVID-19.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? And how would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gather just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane. Now, with the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon. And when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. In a healthy body, hair-like cilia lining the tubes constantly push the mucus and germs out of your airways, where you might expel them by coughing. Normally, cells of your immune system attack viruses and germs that make it past your mucus and cilia and enter your alveoli. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs. Pneumonia may cause difficulty breathing, chest pain, coughing, fever and chills, confusion, headache, muscle pain, and fatigue. It can also lead to more serious complications. Respiratory failure occurs when your breathing becomes so difficult that you need a machine called a ventilator to help you breathe. These are the machines that save lives and that medical device companies currently ramp up production for. Whether you would develop these symptoms depends on a lot of factors, such as your age and whether you already have an existing condition. While this all sounds scary, the push to develop a coronavirus vaccine is moving at high speed. Studies of other coronaviruses led most researchers to assume that people who have recovered from a SARS-CoV-2 infection could be protected from reinfection for a period of time. But that assumption needs to be backed by empirical evidence, and some studies suggest otherwise. There are several different approaches for a potential vaccine against the coronavirus. The basic idea is that you would get a shot that contains faint versions of the virus. The vaccine would expose your body to a version of the virus that is too weak to cause infection, but just strong enough to stimulate an immune response. Within a few weeks, cells in your immune system would make markers called antibodies, which would be specific for only the coronavirus, or specifically its spike protein. Antibodies then attach to the virus and prevent it from attaching to your cells. Your immune system then responds to signals from the antibodies by consuming and destroying the clumps of viruses. If you then catch the real virus at a later stage, your body would recognize it and destroy it. In other words, your immune system is now primed. Collecting evidence on whether this will be possible, safe, and effective is part of what's taking researchers so long to develop a vaccine. It's a race against time to develop a vaccine amid a pandemic. Each step in vaccine development usually takes months, if not years. An Ebola vaccine broke records by being ready in 5 years. The hope here is to develop one for the new coronavirus in a record-breaking 12 to 18 months. While all this will take time, stay home if you can, to protect the most vulnerable. And don't forget to wash your hands for at least 20 seconds and as often as possible. This video
How about the symptoms of COVID-19? You can develop symptoms anywhere 2 to 14 days after you've been exposed to the virus. Typically, people develop symptoms within 4 to 5 days. Symptoms may include a runny nose, sore throat, fever, cough. You may have difficulty breathing, extreme fatigue. You may have myalgia or muscle aches. You might have a headache. You might have anosmia, which is a loss of smell. You might also have GI symptoms like vomiting and diarrhea. According to the World Health Organization, about 80% of the people who get COVID-19 will not require hospitalization and should recover within about 3 weeks' time. What complications can result? About 1 in 6 people become seriously ill and develop complications. These complications can include things like pneumonia, acute respiratory distress syndrome, blood clots have been seen, there could be injury to the heart, and children are also at risk for rare complications referred to as pediatric multi-system inflammatory syndrome. Many of these complications are caused because of a condition called a cytokine storm. This is when the infection triggers your immune system to flood your bloodstream with chemicals called cytokines. Cytokines are anti-inflammatory proteins, are inflammatory proteins. And so cytokines can kill tissue and they can damage organs like heart, kidneys, and even your bladder. All right. I've already told you about modes of spread. So in the interest of time, I'm going to sort of skip over this really quickly. You know that it's a respiratory virus. You can get it by close contact, or you can get it with respiratory contact with respiratory droplets, or direct or indirect transmission by touching surfaces or objects that have the virus on them. How did this outbreak evolve? So this here shows you a timeline of some of the early stages of the outbreak. So like I said before, by late December, the number of cases of pneumonia were increasing in Wuhan. By 29th of December, 4 of the cases of this unknown pneumonia had been linked to a local vet market, Huanan seafood market. By December 31st, the CDC in China informed the World Health Organization about this unknown pneumonia due to unknown causes. Within 7 days, the causative agent had been isolated. They had figured out it's a virus. They figured out it was a coronavirus, and it was named SARS-CoV-2. Just 3 days after that, the entire genome had been sequenced, meaning the entire RNA, the genetic material of this virus had been sequenced, speaking to the explosion that has happened in the field of genomics. By the 3rd week, Wuhan was under a complete lockdown, right? And by January 30th, WHO had declared it as a public health emergency of international concern. Within a month, by the end of February, COVID-19 had spread to 47 countries and more than 82,000 people had contracted COVID-19. I'd like to show you a graphic now that shows how quickly this outbreak became an epidemic and then a pandemic. So as of today, there are over 18.4 million cases of COVID-19 all over the world. The size of the circles here show the countries that have been most affected. And these are the US, Brazil, and India. So how do you predict the spread of an outbreak? Researchers and mathematicians all over the world have developed countless epidemiological models so as to project how this epidemic will spread. How did it become a pandemic? Where is it going to spread? Some of these models also incorporate different public health policies that have been put into place. Things like masking, social distancing, and stay-in-place orders have all been factored into these models. One of the metrics that they use for these models is referred to as R0. R0 is the reproductive number. This is the average number of people who can be infected by one person who has COVID-19. If R0 is larger than 1, then the number of infected people goes up exponentially, and you're going to end up with an epidemic. If R0 is less than 1, then the outbreak is going to die off on its own. So R0 is like an early warning system. It can provide a measure of how fast an outbreak can spread. So let's look at this graphic over here. This compares the R0 for different diseases. So if you see in the bottom panel, it shows you seasonal flu. This is the influenza that we get every year, right? The R0 for it is 1.3, which means one person with flu can spread it to a little more than one person, maybe 1.3 people on average. In the middle panel, you can see measles. Measles is a very contagious disease. It has a high R0. The R0 for measles is estimated somewhere between 11 and 18. That means one person who has measles can spread it to 11 or more people, right? SARS-CoV-2 is not as bad as measles. It's a little worse than the seasonal flu. The R0 for SARS-CoV-2, shown at the top there, is estimated somewhere between 2 and 3.1, meaning one person can spread it maybe to 2 or 3 other people. And this is only a preliminary estimate and is likely to change. But R0 is an effective metric only at the beginning of an outbreak because it assumes that the entire population is susceptible. A more effective metric that is used is called RE, or effective reproductive number. Because what it does is it takes into account some of the mitigating measures we use, that we are masking, that we are social distancing, that we, in certain places, there are stay-at-home orders. All of those are factored into RE. This here is from the Johns Hopkins COVID dashboard. And this is a graph showing you how the outbreak evolved in the 10 most affected countries. 6 of those countries are in the American subcontinent. These countries also have a population of more than 1 million. So what is plotted here is the incidence or the confirmed new cases over a period of the last 7 months, right? Or 6 months. And you can see the green line represents the US. You can see that the outbreak took off somewhere in late March. We were having over 30,000 cases. And then various regions started putting mandatory stay-in-place orders. And then by May, we started seeing an effect. The number of cases were going down. People were social distancing. They were wearing masks. So our numbers were down a little bit to about 20,000. But then what has happened in the last 5 or 6 weeks is really alarming because a number of places have started opening up probably before they should have. Some people are not social distancing. Some people are not masking. And so we see that the number of cases spiked up to about 70,000 cases every day in the US. And it sort of hovers right between 60 and 70,000. Another country that is affected a lot is what is seen in the orange line there, that is Brazil. There are over 2.5 million cases in Brazil and over 90,000 deaths. India is another country that is affected largely is affected a lot by COVID-19. The line for India there is seen in pale pink, and you can see how that line is going up exponentially. None of us ever thought we would be living through a pandemic. But if you look at the history of the world, pandemics are not that unusual. A pandemic is defined as an epidemic that occurs worldwide or over a very large area that crosses international boundaries and affects a large number of people. On March 11th, WHO declared COVID-19 a pandemic. Today, there are over 18.4 million cases in over 188 countries, and 4.7 million cases are in the US itself. How about closer to home? This looks at Michigan. And so what you're seeing here are 3 different metrics: the number of new cases, the testing rate, and the percent positivity. And what you're looking at is the 7-day moving average. So for the top graph there, it shows you that there were 616 new cases today. And you want the line to be green because that represents a day-over-day decrease in cases. Right? If the line is red, it's day-over-day increase. And you can see that for this week, we're trending up, which is not great. We're doing about 3.2 tests per 1,000 people, as seen in the second graph. The good news is that our line for the past 7 days, we are trending up. That means we have a day-over-day increase in average testing. Our percent positivity, meaning of all the tests we do, how many are positive? Our percent positivity is good. You want the percent positivity to be below 5%. And we have hovered all the way right around 2.4, 2.3%. Right? So that means we are testing, we're casting the net wide enough. We are testing enough people that our percentage positivity is below 5%. Quickly talk about the diagnosis of COVID-19. There are two tests. Primarily, the viral test is what is being done primarily. You take a nasal swab or a throat swab, which I'm told is not very comfortable. The healthcare worker collects fluid, and what is done is a modified PCR, a polymerase chain reaction, which detects the genetic material of the virus. You can get results back in 1 or 2 days, but it may take up to a week or two. Their turnaround times are longer now. The newer test is not as widespread. This is the antigen test that is testing for certain proteins on the virus. This sample collection is very much the same as the viral test. The antigen test is cheaper, and also you can get the results back sooner, which is why it might be more practical for larger numbers of people. Now, I want to give you a brief update on vaccine development. Vaccine development has been going on at an amazingly fast rate. There are, let me tell you about 3 vaccine candidates. The first one is being developed at the University of Oxford with AstraZeneca. This vaccine is called AZD1222, and they have already completed what is called as their phase 1 and 2 trials for this vaccine. They found it was safe and it was stimulating an immune response. They had both antibodies and T cells. This has gone into phase 3 in Brazil and South Africa and in parts of England. And AstraZeneca said that they could make up to 2 million doses of this vaccine if all goes well. Let's talk about the 2 vaccines being developed closer to home. One is a Moderna vaccine, the other is a Pfizer vaccine. Both of these are RNA vaccines, so they are using RNA against that codes for the spike protein. So the idea is that they would give you this RNA, your body will make the spike protein on and display it on your cells, and then it would evoke an immune response and you would make antibodies. Both of these vaccines have moved into phase 3 trials, and they're enrolling about 30,000 volunteers. So we, like Dr. Fauci said, we are cautiously optimistic that we could have a COVID vaccine by early 2021. I'd like to end with a cautionary note. This pandemic has been devastating. But if you look at the history of the world, pandemics are not that unusual. Right? What is happening in the US now is concerning because it looked like in May we had some control of this pandemic, but now the numbers have spiked up quite a bit. So how do we get control of this epidemic in the US? So these are recommendations made by expert epidemiologists over at Johns Hopkins. They say that we have to perhaps encourage and mandate masking, social distancing, which are your non-pharmaceutical interventions. You want to close high-risk activities like bars in areas where the epidemic is worsening. You want to bolster PPE supply chains and test supply chains so that test turnaround times are also shorter. And finally, you want to plan for a vaccine, not only producing it, but also its distribution and making sure that the community is engaged and that there is less vaccine hesitancy. Thank you very much for listening to my talk. I would be happy to take any questions. Please stay safe.
Okay, thank you so much for that wonderful presentation. And now we will move into the question and answer part of our evening together. And we got a lot of questions that came in, so we'll get to as many as we can. So first and foremost, can you talk about, and I think many of us have a general idea of this, but can you talk about who is at risk or most at risk of developing complications from COVID-19?
So it's thought that about 20% of people who get COVID-19 end up in the hospital. And a lot of those people develop serious complications. So who's at risk? Mainly older people, people over the age of 65, people who have underlying medical conditions, like who have cardiovascular problems, lung disease such as COPD, who have diabetes, who may be obese, people who are immunocompromised. Maybe they are fighting off HIV or they are taking drugs like steroids that suppress their immune system. All of those people would be at higher risk at developing complications from COVID-19.
Okay, next question. So normally it takes years to develop and test vaccines before they can be put to use. So can you talk about how has that changed and why are people hopeful and expecting that a COVID vaccine would be ready in early 2021?
Yeah, normally vaccine development is a lengthy process. It can take anywhere between 7 to 10 years, and it is fraught with failure, over 93% failure rate. The thing that has happened with this vaccine, the reason we can say that this vaccine may be produced in 12 to 18 months is because of several things. Over the last decade, scientists and vaccine developing companies have been pushed by outbreaks of Ebola, outbreaks of H1N1 influenza to shorten that vaccine production time. And so an Ebola vaccine was produced in 5 years. That itself was regarded as short. Many of the people, the 3 vaccine candidates that are being made right now are being made by scientists who have expertise in SARS and MERS, other coronavirus outbreaks. So they knew what worked. So the research time was cut down quite a bit. So, and the other thing they're doing is they're combining phases of the vaccine development, phase 1, phase 2, phase 2, phase 3, so that they can shorten that time. And so the fact that there are 3 vaccine candidates in phase 3 trials is very encouraging, and we hope to have a vaccine by early 2021.
Okay, our next question. So what are the treatments that are currently available, if any, for COVID-19?
So a lot of the treatment for COVID-19, if you go into the hospital, is just symptomatic care, supportive therapy, right? So part of it would be giving you oxygen, putting you in a prone position, giving you oxygen either directly or through a ventilator. Complications of this disease is coming from systematic inflammation all over your body. So they might give you a combination of anti-inflammatories, such as dexamethasone. Also, they might give you blood thinners so that you do not develop blood clots. The only antiviral that has been approved for treatment of severe patients of COVID is remdesivir. So remdesivir shortens the recovery time from about 15 days to 11 days in critical patients.
Okay, so is there a possibility that the virus strains have recombined and evolved over time? And if so, what does that mean in terms of vaccine development?
So a lot of the major recombination happens before it jumps to humans. Now that the virus is already in humans, the only way it can change is by mutations. And coronaviruses don't mutate as much. They don't mutate as much as even the influenza virus. So the reason part of the reason we need a new flu shot every year is because the influenza virus mutates so much. in humans, coronaviruses can mutate, but they don't mutate as much. And so the expectation is that the vaccine should be useful to us.
Great. So this is an interesting question. Why are first stage reservoir and second stage reservoir species not badly affected by coronaviruses, but humans are severely affected?
So for some reason, bats seem to be this sort of incubator, and they have known to have several species of coronaviruses, and the bats do not get sick at all. Part of it is to do with their metabolism. They have a very high rate of metabolism, and for some reason, their immune system does not get triggered by these viruses, so they kind of live in equilibrium with these viruses. Some of the intermediate hosts, some of the camels, which were suspected of transmitting MERS, they did have mild disease. So it's possible that, so the question is, if your immune system doesn't react, then you are not going to provoke, you're not going to get any sort of symptoms.
So in your opinion, why are the USA, Brazil, and India really outliers with regards to the spread of the virus? What is it about these three countries that has led to higher rates than in other countries?
So I don't think there's one or two factors that can really point to this. Part of the problem is that, especially in the US, is that we have opened too early, that we have not sort of kept our mitigating, whatever we can do to mitigate transmission, all those lockdowns in place much longer. Even who said we have not followed the guidelines that were set out by CDC and World Health Organization. And so, and then you keep hearing about people having large parties and not wearing masks. And so you see these cases creeping up. Well, the demographic has changed in the US. More and more younger people are getting COVID now than were getting it in March and April and May. So it's probably because these younger people are out much more and getting the disease. I do not know that much about Brazil and India. India was initially quite effective in locking down the entire country. And it's also hotter in India, which is why it's very surprising that this virus has spread, because initially they kept telling us that heat would kill off some of these respiratory viruses. So it's not completely clear why there's so much community spread in certain areas.
So if it is true that we get a vaccine in the next year, and I really like this question personally, but is it possible, can we have hope that a vaccine for the common cold is coming?
So the thing about the common cold is there are over 200 different viruses that can cause the common cold. Some of them are rhinoviruses, some of them are coronaviruses, some of them are para-influenza viruses. And probably the common cold is not severe enough to justify spending those, $500 million that you seem to need even to make a vaccine against one strain, forget 200 different strains. So I doubt that is going to happen.
Well, that's disappointing. So how does an immune system store knowledge of a virus? It's seen before and transmit that information throughout the body. So is the DNA or RNA code stored somewhere and then compared to incoming viruses to be aware of possible threats? Or how do antibodies know which viruses to adapt to?
So there's been a lot of focus on antibodies, but actually our immune system has a lot more tools in its toolbox than just antibodies. So you start off in your immune system, it basically has two arms of your immune system. One is what is called as innate immunity. That's what we have all the time. It's non-specific. Those are your barriers that you have, your physical barriers that you have, your skin, your cilia that are beating and so on. And then you go on to your specific immune system, which is your adaptive immune system. That itself has two kinds of cells. There are B cells and T cells. When a B cell gets exposed to a foreign invader, an antigen, it produces antibodies. The antibodies can bind to the agent and alert other parts of the immune system to eat it up. When that B cell makes contact with the foreign antigen, there are memory B cells that are made. So it has these cells that last in your bloodstream. Antibodies may only last for a few months or maybe a year at most, but it's those memory B cells that you have, and there are also T cells, on the other hand. You make memory T cells. It is those memory B and T cells that we keep for years, if not for our entire lifetime, to protect us against various diseases. So whenever we see that pathogen again, the memory B cell will produce much more antibody much more quickly.
So we're hearing a lot of talk about the possibility of a second wave of coronavirus in the United States. And so if that were to occur, will that look different than the first wave? And if so, how?
Well, the first wave has been completely unpredictable. We thought we had some control over it, and then it's starting to trigger up again. So I think this virus is very, very unusual in the extent of community spread, in the fact that asymptomatic people can spread disease, in the wideness of its clinical manifestations. So ideally, when you get a second wave, your first wave died down, so a lot of the people should have been exposed to the disease and maybe have partial immunity at least. So the hope is that if there is a second wave, that it should be less severe on us.
So you mentioned the term rhinovirus. Can you talk about what that is and how is it different from a coronavirus?
So a rhinovirus differs because, for one thing, it is what is called as a naked virus. It doesn't have that outer envelope like the coronavirus has. It also has different proteins that it displays on its surface, or different antigens it displays on its surface, by which it would interact and stimulate our immune system. So it belongs to a completely different family of viruses altogether.
Okay, so there was research on vaccines for other coronaviruses. Are they using this research then to help support a vaccine for this coronavirus?
Absolutely. So the fact that we have cut down our vaccine development time is mainly because the scientists in some of these trials are scientists who have expertise in starting to develop a vaccine for SARS and MERS. Finally, vaccines were never developed for those two diseases. They were just in development. And so they are using the same ideas. So they knew that they could use the spike protein, that it would stimulate an immune response. And so they are taking their pointers from those vaccine developments.
So why do we hear about some people getting very sick and even passing away from COVID-19? And yet conversely, you talked about 80% of people not even needing hospitalization. So why do some get really sick and others hardly show any symptoms at all?
So doctors have been really puzzled by the extent of its clinical manifestations that we see in COVID, asymptomatic, mildly symptomatic, really severe, people who are so sick, people who die, people who have, lingering symptoms for months on end. And we don't completely know why this is the case. I've already told you a little bit about who's at high risk. So those people who are at high risk, the older people, people with underlying medical conditions, those would go on to have more susceptibility to some of these complications, for sure. But why are some people asymptomatic? There were some clues in a couple of research papers that were published recently. These people were surprised because they were looking at blood samples of people from 2015, way before this coronavirus hit. And they found that those people's blood samples had T cells that could recognize parts of this coronavirus. So those people are coming in with some unknown level of protection against the coronavirus. And why? probably because they were exposed to the other 4 coronaviruses that cause common colds in millions of people every year. So there is definitely a fraction of the population that is coming in with some unknown level of immunity. And that might explain why somebody, instead of going to the hospital, just gets sick and is able to manage at home, or instead of getting moderately sick, if they had this level of immunity, they would not get sick at all.
So would it be possible for some of us to see another virus as widespread as COVID-19 within our own lifetime?
At this point, so many rules have been thrown out the window that anything is possible. If you had asked me 3 or 4 years ago whether we would see a pandemic that was this widespread, I would have disagreed vehemently with you. I think there's one YouTube video of Bill Gates who had said that the world was not prepared for the next pandemic. And that was largely true. I think we were not prepared for this pandemic. Let us hope that, devastating though this pandemic has been for the whole world, that we learn, different nations ramp up and are more ready for something that might emerge in the future.
Well, you walked right into our next and final question perfectly, which I had been preparing as our last question, but you really segued nicely here. What can we do as a human species to be better prepared for a future or next pandemic?
So I guess things would have to be done at several different levels, like federal, state, local. The idea is that you have more budgets for the public health departments. Especially in the US, we don't do as much contact tracing as we need to. So contact tracing is an important part of epidemiology. Whenever an outbreak starts, and I think Dr. Droust was talking about how he was a contact tracer for tuberculosis last week, and he hopes to do some contact tracing for COVID as well. We don't do as much contact tracing in the US. What is contact tracing? If someone has COVID, you're trying to find out who is that person come in contact with and helping them all isolate for 14 days. You want to break that cycle of that chain of transmission. In Florida, it's estimated that you needed about 300 contact tracers per 100,000 people, and we have only 7 contact tracers per 100,000 people. So that just tells you that we need to invest in some of these resources in surveilling, in doing more surveillance of diseases so that we can we can put out the fire much before it becomes a blaze.
Okay, and with that, folks, we are halfway done with our 6-part Pandemic Perspective series. I do want to spend, as we do each week, a couple of moments going over some brief housekeeping details. As a reminder, the link you used to access tonight's presentation will be the link you continue to use for the remaining 3 weeks. All our presentations are recorded, and those recordings are available on our website within 24 hours of the presentation. And in the Canvas site, students will find a 5-question quiz. Please be sure to take the quiz no later than next Monday night at 11:59 p.m. And as a reminder, again, to those earning credit, you must participate in the sessions, complete the quizzes, and complete the reflection assignment, which will be given at the end of our 6 weeks together. Lastly, I just want to invite all of you, for those of you specifically not joining us for credit, to stay with us again next week. Next week, we will be joined by Dr. Brenda Whitehead, who will discuss the impact of COVID-19 on multiple aspects of life for older adults, focusing on factors associated with higher risk for stress and mental health issues, and behaviors or resources older adults most report as helping them maintain well-being in the midst of stress, framed within a lifespan, developmental, psychological perspective. So do join us. Thank you again for being here this evening, and special thanks again to the team who's been working diligently for weeks to put this series together for you. Have a great week. We will see you next Tuesday. Thank you.